everybody. Welcome to our first podcast in our information domain. In this domain, we will spend a lot of time with DNA, biotechnology, and inheritance. So let's dig right in because DNA. So big questions. What's it look like? How's it work? And how was the initial structure of DNA determined? So we're going to be having a little history lesson here. So first and foremost, we're heading back a century to really figure out what was known. So by the middle of the 20th century, genetics was pretty well established. We understood that things were inherited thanks to the work of Mendel in the mid-1800s. We understood about inheritance, but what we didn't know was how genes worked, what they were made of. Nobody really knew that answer. So DNA's role in inheritance in that discovery is probably the most significant contribution to our understanding of how life works. And, you know, we only recently celebrated the 60th anniversary of the structure discovery. So, you know, it's still new, and yet we've come so far. So this understanding has not been the sole piece of any one single person's work, but many people's work. So that's what we're going to explore here, is who were the key contributors to that. So just to say what I just said there, you can stop and go back and write that stuff down. Friedrich Meister. He is who we credit with the actual discovery of nucleic acid. He first looked at a nucleus, found that there inside the nucleus there was an acidic component, and called it nucleic acid. Okay, it makes sense. 1869. All right, so we get those. Then we start noticing that there are some chromosomes. Okay, during cell division, it was noticed that we had the chromosomes. They were a way of duplicating, pulling apart. Two ingredients to chromosomes: DNA, protein. Problem was, which carried the heredity information, DNA or protein? Big divide in the old scientific community during this time. Before 1940 or so, people thought it was protein. It was a big division. Some people were in the DNA camp, but we didn't know its structure. We only know what it was made out of. We didn't realize, you know, people just weren't putting the pieces together yet, and we thought it'd be protein. Protein's far more complex. It's far more interesting. And so we didn't know about the structure yet. So that's why we weren't sure what the heredity information was. So in 1928, we get Frederick Griffith. Okay, Scottish microbiologist. His big thing was that he was looking ultimately for a vaccine for pneumonia. And what he ended up discovering was the fact that bacteria could give other bacteria heritable traits even after they were dead. Whoa. Okay, so his work, not intentionally, helped lead the way towards helping us understand that DNA was the actual inheritable material. But that's where he left it. He didn't care. He didn't care that that was going to be his contribution. He wanted to find a pneumonia vaccine after watching so many soldiers die during World War I. So his study was wrapped around that. And again, inadvertently, he helps to pave the way for our understanding that DNA was actually the inheritable information. So he studied two forms of pneumonia. There was a rough kind, that's the one on the left here, very harmless, not going to cause any problems, might make you sick, not going to kill you. The smooth kind, though, the one on the right, very pathogenic, is going to kill you. So he does his studies in mice, and we've seen this in the past, so here's just a reminder. Feel free to pause to take a look, because I'm just going to kind of zip through this real quick. Basically what he did in his experiment, the way that he designed it, he took living smooth as his control, injected the mice. That's it. Mice dies. Yep, makes sense. It's pathogenic. Takes the living arm. The rough kind. Yep. Mouse lives, okay, because it's non-virulent. Now, in order to find a vaccine, generally what you do is you inject some form of the original, whether it's dead, remnants, whatever it is, in order to have the body build an immune response. So then he takes heat-killed S. The mouse is healthy. Makes sense. He killed the bacteria and injected that into the mouse. Cool. So now let's start to build up a vaccine. So he takes the dead, ones that make the mouse healthy. He takes the living R, again, healthy mice, mixes them together into the syringe, and injects those. Now, given the fact that the living R, the mice is fine, are fine. The heat killed S, the mice are fine. One would imagine combining the two, the mice are going to be fine. Okay, except when he did it, mice are all dead. Okay, does little autopsies, finds inside the mice now, not living R, not dead S, but living S cells. What? Okay, so here's the question. How the heck did that happen? Turns out he found a transformation factor that pieces, the DNA, the inheritable material, was being taken up by the living, okay, non-virulent kind. And when they divided, they now had the genes to make the nasty coat, makes the smooth coat, and they're all living R. Avery then, we move now to the 1940s, refines the experiment, redoes the experiment, but instead of doing it inside mice, he does it in a test tube and finds that the same results, okay? Yet, haters still existed. No way could it be DNA. So he figures out that DNA is what's being picked up, basically being picked up, this transformation factor that's being picked up by the living R bacteria, and they're using those genes to create a smooth coat. I mean, really, how do you not be okay with that, right? No, not good enough. Okay, in come Hershey Chase. Now we're just before, okay, 1952. 1953 is the big year. That's when the structure of DNA is found. This is going on at the same time. We get Hershey Chase's experiment that's going to put the nail in the coffin, okay? All right. They are working with bacteriophages. They're doing this at Cold Spring Harbor Lab in Long Island. Alfred Hershey, Martha Chase say, you know what? We want to figure this out. Let's do it, okay? We want to know if it's protein or DNA. Let's use something that only has protein or DNA. We're going to use a bacteriophage. Okay, these are viruses that only infect, okay, bacteria. Okay, so what they do. Okay, here's what they do. They take the bacteria. They're going to the virus. They're going to radioactively label the protein coat using, let's see, what element would protein have that isn't found in DNA? We have to remember these things, right? Well, okay. Remember, protein, carbon, hydrogen, oxygen, nitrogen, sulfur, protein, right? The big elements. DNA, carbon, hydrogen, oxygen, so far the same. Oh, nitrogen, oh, shoot, phosphorus. There's the big difference. Sulfur in protein, phosphorus in DNA. Let's radioactively label each one, and we'll track it. Okay, let's find out where it goes. So here's what they did. That's what they did. Okay, let's radioactively label the protein. Okay, when they do that, put it in, shake everything off, okay, and allow the bacteria to then reproduce. They find only radioactivity in the pellet outside, nothing inside the actual bacteria. When they use the phosphorus, radioactively label the phosphorus, they do the same thing. Boom, shake, 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 shake. They find only the radioactivity inside the bacteria, which means that's the inheritable piece, because that's what the phage injected into the bacterial cell to make new bacteria, or to make new virus. So the only way new virus is made, okay, is from that DNA being injected inside the bacteria. Aha, boom, done. Question answered. Okay. At the same, can you imagine living during this time? I just got to back up. Seriously, like the 1950s, early 1950s, when this is happening, you got Hershey Chase going on. We got Erwin Chargaff figuring out his rules, which demonstrates how DNA is actually composed. We knew what it was made of, okay? Now we've got Erwin Chargaff figuring out how things are kind of found in particular ratios, right? So this Austrian biochemist figures out the major important rules of the composition of DNA. And he does it by taking ratios of the 4 main nitrogen bases. Okay, so we know that DNA has 4 bases: adenine, guanine, thymine, cytosine. Okay, so when he found this, even though each species he used have different amounts of these materials because they have different DNA, right? No matter what he did, though, the ratios were nearly identical of adenine to thymine and from guanine to cytosine. It didn't matter what the species was. Those were always the same. The amount of adenine was equal to the amount of thymine, the amount of cytosine equal to the amount of guanine. Why would this matter, right? Why would that matter? This is, again, building between Hershey-Chase's information. This is now starting to feed into Watson and Crick and what they're doing, and they know about these rules, okay? And this is going to help them start to figure out we're dealing with a double helix here. And you can check out this link that just kind of animates the idea of Chargaff's ratios here. So if this is still, if you want a kind of a visual, check this out. So now it's 1953. Now, obviously, this didn't happen overnight. For a few years, okay, Watson and Crick, Franklin and Wilkins are all working on this problem. Not to mention, you also have Linus Pauling, a U.S. guy who's already won the Nobel Prize, ends up winning 2 of them, figuring this out. Oh, let me go back too. Hershey Chase, they win the Nobel Prize down the road for what they did with their work on bacteriophages. That's how important, like all of these, I mean, these are nearly all Nobel Prize winning discoveries. So you got Watson and Crick, okay, these two competing teams, even with Linus Pauling also thrown into the mix here, trying to figure out the actual structure now of DNA. So here are our 4 main players, okay. One's working at Oxford, one's working down the road basically at King's College. Watson and Crick, you know, basically take the X-ray crystallography, the X-ray diffraction that Rosalind Franklin takes, that wicked important, super well-known X-ray picture that you guys see here, to help them determine that what we're dealing with from a molecular structure standpoint is a double helix. Okay, so important. So between that, the ratios of Erwin Chargaff, okay, having all those pieces, knowing what went together, Watson and Crick figure out that it's a double helix, sugar phosphates on the edges, the bases coming into each other, and each base pairing properly with the other one. Adenine always binding to thymine, guanine always binding to cytosine. For their work, one of the fastest turnarounds in Nobel Prize history, within 9 years. They win the Nobel Prize. Sadly, Rosalind Franklin does not. She passes away in 1959 from cancer, so she does not officially get named as one of the recipients, but we don't argue her contribution to this. It's too important. So there it is. We know it, love it, live it. Our DNA. Important little molecule, isn't it? Yeah. That's cool stuff. So, first puzzle solved. We now know DNA is the molecule of heredity. So what the heck and how the heck is this thing structured? Okay, here we have it. Here it is, kind of unwind. Take a second, make sure you draw it out, make sure you know the pieces. Okay, you've got your 5' end. It runs in a 5' to 3' direction. That prime piece deals with the location of the carbons. Okay, so each of these is a carbon at each vertice, right, of the ribose sugar. Okay, this is carbon number 5. You start with carbon 1 in organic chemistry language. You work your way around clockwise. Here's carbon 1, carbon 2, carbon 3, carbon 4, carbon 5. So carbon 3 sits down here. So 5 is above the 3. Think of your house. Peak of the house always points up in the 5' end. On the other side, you've got the 3' end up, because there's my third carbon. I'm still going here because it's upside down. 1, 2, 3, 4, 5. Now my 3 sits above my 5. So they run what we call anti-parallel. One's right side up, the other one's upside down. Okay? A nucleotide, the actual monomer, is the phosphate, the sugar, and the base attached. That's how they're put together. Here's my T, opposite my A. Okay, so you've got your purines and your pyrimidines, just groupings, the way that we group them. Guanine, okay, and adenine are purines. Your pyrimidines are cytosine and thymine. So purine always binds to a pyrimidine. And you can see the reason why they bind so perfectly is because there's hydrogen bonds that attach. And A to T only create 2 hydrogen bonds, while G to C create 3. Okay, so we have a couple of things. Here's just the way the structure works, is that double helix, showcasing the bases. Here's the actual partial chemical structure, the way that it's actually put together, and here's a space filling model. Pretty cool, okay? So bases on one strand are covalently bonded to each other, the phosphodiester bonds. They're held together super tight by covalent bonds. Opposite strands, so the two sides together, are hydrogen bonds. That makes it tight collectively, but easy to break for things like replication. So chromosomes are just densely packed double-stranded DNA with hundreds of millions of base pairs, each chromosome of different length based on the number of base pairs that make it up. And then you have chromosomal proteins that help with mediate how they're packed and how they're actually put together. So, if we look at here with the various scaffoldings, we start here, there's the double helix, okay, that's about as close as we get. We have seen it's actually been seen now. We've gotten that close where you can actually see the twist. Okay, so there's your single there, okay. As we start to bring in histone proteins that wrap together, okay, the DNA starts to wrap around the histone protein twice. That's called a nucleosome, okay. So constant wrapping around that. Then we get those wrapping around those. Let's show you that. Those wrapping around those. Packing, denser, and then those loops wrapping around those loops, and then you get your chromatid. Okay, and if you have a replicated chromosome, that's called, one is the chromatid, the other one's its sister, so we'll talk about that, and this is what you would see during cell replication. From this, make sure you can do all these things. I realize this was kind of quick. Remember to stop if there are things that you still need to review. You have your text to go back and review. Make sure you understand how the structure is related to the information that's stored, who the contributing scientists were, what their contribution was, and how that process led us to where we are. And from here, we're going to dive in replication, the central dogma, how we go from DNA to protein, protein gene expression, understanding prokaryote versus eukaryote, gene expression, biotechnology, and ultimately how we inherit the traits we do. So have a great evening, and we'll catch you later, guys.